In this video, I will describe the unfortunate incidents that led to the development of Parkinsonian symptoms in a 23-year-old chemistry student and 7 years later, a different group of people known as the Frozen Six. In 1973 in Maryland, USA, a chemistry student called Barry Kidston was sneaking lab equipment out of his college lab, looking for legal ways to create recreational drugs similar to heroin. He had heard of a paper by Ziering and Lee from 1947 that successfully synthesized desmethylprodene, or MPPP, which is an opioid analgesic with similar effects to others in the class, but with fewer legal restrictions. After a bit of research, Barry began to produce his own batch of the drug, and within months, he developed symptoms of Parkinson's. Doctors were completely stumped, as when they treated Barry with Parkinson's treatment levodopa, a precursor in the dopamine synthesis pathway, which allowed some of the undamaged neurons to bounce back. It relieved his symptoms. But Barry was way too young, and his symptoms had developed way too quickly for it to be Parkinson's. Even after further investigations at the time, there were no breakthroughs, and testing of byproducts of this synthesis in rats also proved to be in vain. Fast forward to 1983 in California. A patient presents to the hospital showing typical symptoms of catatonic schizophrenia, such as waxy flexibility, where a person's limbs stay where they are after being moved by another person. Dr. Langston, a prominent neurologist at the time, was asked to consult, and he did a test to determine if it was indeed catatonic schizophrenia. The doctor moved the limbs of the 42-year-old patient, George, and George's muscles didn't put up any resistance. This is inconsistent with catatonic schizophrenia. What's more is that George's hands displayed cogwheel rigidity, moving in small, jerky movements, a typical sign of Parkinson's. But George was only 42 at the time, and these symptoms developed over just 3 days. Further investigations revealed that George's girlfriend, Juanita, was also in this frozen-like state, but her family had been taking care of her in fear of legal repercussions. This led doctors to believe that something that wasn't above board was occurring. When taking a history, a medical student saw George's hand move slightly, and in an attempt to communicate with George, he put a pen in his hand to see what he could scribble down. When asked about any medication George had taken, he wrote down heroin. Due to the word getting around about these rare, mysterious cases, neurologists in California discovered that there were 4 other patients with similar symptoms, and the only thing in common between them was that they were all heroin users. Evidently, the cause of this incident was the heroin the patients were taking. But this isn't a normal side effect of heroin. At this time, legal synthetic opioids were being synthesized by chemists that had similar effects to heroin, but due to their slightly different structure, were legal. The problem with these drugs is that their safety is extremely unknown. To examine whether this was the case, Dr. Langston needed to get a sample to test. Once they got a sample and tested it using a mass spectrometer, they began to cross-match the data with the government database. But this search was in vain, until a toxicologist who became entranced with the case said it reminded them of the equally peculiar case in the 70s with Barry Kidston, which had been a result of IV injection of meperidine analogs. There was a chemical that was present in both cases and required further study. The drug that Barry Kidston and the Frozen Six had been injecting was MPPP, and it runs the risk of producing a side product, MPTP, 1-methyl-4-phenyl-1,2,3,6-tetrahydropyridine. The intermediate tertiary alcohol is liable to degradation under acidic conditions if the reaction temperature rises above 30 degrees C. Kidston did not realize this and esterified the intermediate with propionic anhydride at an elevated temperature, consequently forming MPTP as a major impurity. MPTP is chemically similar to dopamine, a neurotransmitter involved in movement and reward circuitry. MPTP is also lipophilic, so it can cross the blood-brain barrier easily. The life cycle of dopamine is very controlled, using enzymes like monoamine oxidase, MAO, and catechol O-methyltransferase to break it down so it doesn't persistently activate dopamine receptors. But due to MPTP and dopamine's structural similarity, the MAO-B isoform on astrocytes tries to break it down as well. This reaction converts the relatively unproblematic chemical MPTP to MPP+, which is a substrate for the dopamine transporter on the membrane of neurons. Once MPP+ enters a neuron, it accumulates in the mitochondria, which is the main source of ATP production in the cell, key for the cell's energy. Here, MPP+ inhibits complex 1 of the electron transport chain and oxidative phosphorylation. This inhibition reduces ATP production in these dopaminergic neurons. As a result, there is not enough ATP to fuel the sodium-potassium ATPase, which is key in the maintenance of concentration gradients. Fundamental to cellular life. With this, the dopaminergic neurons begin to die. Due to MAO-B's pathological role here, administering MAO inhibitors can be used to counter the MPP+ effects, but they cannot reverse any neuronal cell death. This begins to explain the previous diagnoses. Firstly, dopamine dysfunction has been linked to catatonic schizophrenia, producing its hallmarks. Secondly, in Parkinson's patients, we see the destruction of the substantia nigra, the main dopamine-producing neuron center in the brain. The dopamine transporter scavenges for excessive dopamine in the synaptic spaces before transporting them back into the cells. Even though this property is exhibited by both ventral tegmental area, VTA, and substantia nigra neurons, the VDA neurons are protected against MPP+ due to the expression of calbindin. Calbindin regulates the availability of calcium ions within the cell, which is not the case in substantia nigra neurons due to their high calcium-dependent autonomous pacemaker activity. A silver lining of the Frozen 6 and Barry Kidston is that from this arose a new model to test Parkinson's treatments. In animals, researchers inject rodents or other animals with this chemical to induce a diseased model of Parkinson's, which gives them an opportunity to test their hypotheses. Additionally, as a side note, MPTP is used in industry as a chemical intermediate. The chloride of the toxic metabolite MPP+, called ciprequat, has been used as a herbicide.